Hi everybody, welcome back to Human Anatomy and Physiology at Clackamas Community College. My name is Christina Wallace and I'm your lab instructor. This is the second term of Human Anatomy Physiology Lab, thus the by 232L, and we're going to start out this term talking about muscles. And so this lab report review will cover exercise 12, microscopic anatomy. Exercise 13 is more like organization and classification of skeletal muscles. So how do they get their names? What are their jobs? And then there's a review of body movements in exercise 11 that looks at joints and things like flexion, adduction, etc. And then exercise 14 covers muscle physiology. So you will be looking at data from an experiment that we would have done in class. Where we were able to affect muscle fibers physiologically with different solutions and watch contraction under a microscope. Okay, here we are at the online lab report instructions. So every lab report has these online instructions and then the lab report itself. And so oftentimes it's good to keep this lab report instructions open on your computer because it has links in it that you can go click on videos and stuff. So the first thing that our instructions are telling us to do is look at this diagram on page 186 of your lab book. And so what we're going to be looking at is a longitudinal microscope view, a longitudinal section of a skeletal muscle fibers. And so if you remind yourself, remember back to 231 where you were looking at the different tissue types, and remember that skeletal muscle has these striations, so the dark and light bands of the sarcomeres, which are the contractile units. And then it also has some peripheral nuclei, because these are all contracting in here. It pushes its nuclei out to the periphery. There are multiple nuclei per cell, so those are all features of skeletal muscle that we can see in a longitudinal section. We're going to be able to see different things in a cross section, and so I'll talk about that in a moment. And so the first thing that you're going to do, you're going to look at a video of a teased whole mount skeletal muscle fiber and just kind of take a look at what you can see. And then you're going to compare it to the picture above and to a skeletal muscle fiber that you find on histologyguide.com. The teased one is really pale. It doesn't have a lot of stain on it. And so that's kind of why you're comparing the two. So the picture above here and the picture on Histology Guide, there'll be more structures visible. So you'll be able to see more things. And so here we are at the first page of your lab report. You'll take a look at the teased whole mount video, and then you'll compare it to the figure in the book or to Histology Guide. And you're just going to kind of take a look to see what structural details become more visible on a slide that has more stain. Then the bottom of this page is going to kind of talk about the things that are visible about skeletal muscle tissue in a cross section. And so when we do a cross section of a muscle, we're kind of cutting in the same plane as those striations. So we're not necessarily going to see those. So we can't use that as a defining characteristic to tell that we're looking at skeletal muscle. The things we can see really well in cross-section are the connective tissue wrappings around different parts of a skeletal muscle. And those are called endomysium, paramysium, and epimysium. And so kind of read about those and figure out their placement. I'll talk a little bit about them. And so you're kind of asked about why is skeletal muscle important? It does a lot of important jobs. This B question right here should not just be one thing. There's a bunch of things that go there. And then you're going to look at a couple of connective tissue structures associated with muscles and tell me some information about them. So one is a tendon, one is an aponeurosis. And so kind of think about, and you have to kind of compare those structurally and functionally. So structurally is always going to be about what tissue type is it made out of? What does it look like what is its shape in the body? That kind of stuff. And then functionally is what does it do? Why is it there? What does it connect? Does it have to be strong in more one direction or multiple directions? Those are kind of functional questions. Picture is pretty much just straight out of your book, muscle wrappings, the connective tissue layers. And so I just drew it out for you. You have epimysium as the outermost layer. So this is actually a muscle connected via a tendon to a bone. And so this could be a femur, could be a humerus, I don't know, doesn't matter. This is a muscle, and a muscle is made up of smaller units called fascicles. And so if we pull a fascicle out, that's going to be wrapped with paramycium. And a fascicle is made of a bunch of individual muscle fibers. And so each muscle fiber is going to be wrapped in endomycium. And you'll be able to see this kind of in this kind of longitudinal, spread out format, but also these layers are very visible in cross section. Before I look at cross section, I'll just show you the color coded version. So I kind of added some colors. So here, if we're looking at a muscle in cross section, we can see the muscle fibers, we can see their little peripheral nuclei, and we can kind of see how they're wrapped on the outside. So we can see the layers around each of the substructures in a cross section. That's why a cross section is useful and likely will be how you're asked about these specific layers of connective tissue wrappings. And here's a color-coded picture for further clarity. And so here we are at your online instructions, and this part number 2, that's where we kind of talk about the cross-section layers. It shows you where in your book you can find those things. And then the next part of our lab, we're going to look at a slide of a motor nerve ending in a skeletal muscle. What they call the motor end plate is also called the axon terminal in our lab. So we're going to look at an axon terminal. And here's a slide that you can click on to look at that. There's also a picture in your book. I'll show you in a minute. All right, so here we are at your lab on page 2, and that's where you're asked about the neuromuscular junctions. So you're going to look at a couple of pictures. There's a link in your online instructions to a really good picture that's labeled well, and then there's also a picture on page 188 of your lab manual. These are the pieces of vocabulary that you should be able to identify: motor axon fibers, muscle cells, axon terminals. And then B has you describe what happens in the neuromuscular junction. This is not a quickie one-off sentence answer. There are multiple steps to what occurs at a neuromuscular junction, and I want you to understand all of them. Check out Lab Manual, page 187 for that. All right, so here I've made you a little kind of study slide. So neuromuscular junction. In the green box, I tell you a little story. Okay, the brain sends a signal to your biceps brachii muscle. That's your upper arm in the front to contract so that your arm can bring that delicious fresh cup of coffee up to your mouth. The brain and spinal cord say, I want to drink coffee, and they work together to send an electrical signal to your upper arm. There, the nerve fibers of your nervous system innervate the muscle, and they turn that electrical signal into a chemical signal at a synapse. And then once that chemical signal does its job and delivers its chemical messengers across the synapse, then on the other side of that synapse, the muscle fibers can react accordingly. And so these are the steps that I want you to know. And I have left out vocabulary because I want you to double check in your book, page 187 in your lab manual, to kind of understand these steps. So your action potential reaches your axon blank. What is that name for the little feet of the motor plate? And remember, an action potential uses sodium and potassium, right? So sodium and potassium are kind of entering and exiting cells in the nerve at different times to propagate a signal across a distance. And so once we get to a muscle, all of a sudden we're not dealing with just sodium and potassium anymore. We have some other ions that are going to be in play. So your action potential reaches that axon structure, and then an ion channel will open, and a certain ion will enter your axon terminal, causing something to be released by exocytosis. And so that means little vesicles of something are getting released and then sent physically out into a space to connect with something on the other side. That stuff that is released, there's a particular neurotransmitter that gets released there that diffuses across your synaptic cleft, and it binds to something on the other side. So know what that is on the other side. And then once that binding occurs, permeability of the sarcolemma, so that's the membrane of the muscle, changes. And then something channels open. So now we're done kind of with the neurotransmitter stuff. We're back to the kind of the elements from 2 and 3. That has something to do with that answer. Causing an action. So this is a big major action. So anything I have bolded here means it's kind of like the main steps. And so look at page 187 and kind of try to work that out for yourself. And you can double check with me at a Zoom. All right, so now we're leaving behind kind of the microscopic anatomy, and now we're kind of looking more at the gross anatomy of the muscular system. And so first of all, don't forget this question right underneath classification. Define origin and insertion. So kind of understand what those terms mean in reference to the muscular system. And then B, list 7 criteria used for naming skeletal muscles. And so just beware, in your lab manual, there is a figure that is not about criteria. So just make sure you're reading that you're finding the criteria for naming. And then in this portion, so you're going to tell me 7 criteria. And then you're going to tell me 3 examples, 3 literal, real muscle examples that describe that naming criteria. Muscle names tell you lots of information about them. You know, they tell you oftentimes where they start and stop. So that has to do with like origin and insertion. Whether they're bigger or shorter than a muscle nearby that has a similar name. Etc. So the muscle names are really your friends. You can use them to kind of decode where muscles are in the body. And so some criteria for naming muscles. There are 7 criteria, page 194. I'm going to talk about 3 of them here. You have to figure the other ones out. So muscle size is one way. And so you'll see words like magnus or maximus minimus in a name, like gluteus maximus, gluteus minimus, or adductor magnus. Longus brevis is another indicator of size. Fibularis longus, fibularis brevis. Another way muscles are named is the muscle fiber direction. And so literally the way the muscle fibers are pointing in a muscle can help name it. So abdominal muscles are very common. You'll see fiber direction in abdominal muscles. So a rectus muscle is straight up and down. It's somehow parallel to the body. And then an oblique muscle is somehow, if we imagine a midline kind of right here, an oblique muscle is going to go diagonal. The fibers are going to somehow kind of go diagonal on an angle in relation to the midline of the body. And so external obliques start superiorly at the lateral side, and they go medial at the inferior side. So X to the sex, meaning external fibers point down toward the sex organs. Internal obliques, I'll pick another color actually. Internal obliques start laterally inferiorly, and they go towards the midline and end up towards the midline superiorly. So obliques are some sort of diagonal. And then transverse go across horizontally the plane of the body or the orientation of the body like a belt. And so you'll see some rectus muscles or some oblique muscles in the body. Number of origins is another way that muscles get named. I've given you some examples. And then I kind of walk through what origin and insertion means here. I'll let you take a look at that. And there is some information in your online instructions about where to find classification and stuff. Your lab report goes on to kind of define the main jobs of a muscle. So types of muscles are here. And then we'll go on to talk about body movements. So again, it's just a reminder that you can find these in exercise 11. I've given you the page numbers here. Just kind of understand the different actions of a muscle. And I have highlighted here, include the plane. And so think about the plane of movement. Flexion works kind of more on the sagittal plane, whereas abduction works more on a coronal plane. And so I want you to figure out what I mean by that stuff. And then at the bottom of this page, with your own bodies, demonstrate the following body movements. And so these are all the kinds of movements that in future lab reports, you're going to have to find pictures of muscles doing lots of these things. And so just kind of work through these things and make sure you understand what they do, what the difference between hyperextension and extension is, what the difference between abduction or rotation is, supination, pronation. Be familiar with this vocabulary. All right, and the very last part of your lab report is muscle physiology. And so we're back at the online instructions, and it just walks you through nicely, kind of describes what the experiment is. It gives you a video so that you can watch these different solutions being dropped onto a muscle fiber and see what happens. And if you see, I've highlighted how do the muscle fibers look different before and after contraction. That is a hint that you might need to describe what happens, what contraction looks like in a muscle fiber on a test. So I would have a couple of handy adjectives ready. Think about what occurs and how to describe it visually. And so the first part of this, if we were in the lab, you would be looking at tissue and doing this and drawing some little drawings, but you don't have to draw anything on this page for me. Just kind of be aware this whole page is about watching the video and thinking about what contraction looks like visually so you can describe it. The last portion is you are given data. So all of this data that you see here was created by other students in a lab where they dropped solutions. So a salt solution on one piece of muscle, and then ATP only on another piece of muscle, and then salt and ATP together on another piece of muscle. And these are the findings. And so what you have to do is average the rows across to find an average, and then you're going to be finding the percent contraction. And so you can look in your book on, I think it's page 233 in your lab manual. We'll talk about the 233 muscle fiber contraction experiment. And I've just kind of highlighted for you here how you find the this is how you find the percent contraction. And so you take the initial length minus the contracted length, and then you put that sum, you divide it by the initial length again, and you times the whole thing by 100, and then you get a percent contraction. So that's what I want you to do. Find the averages, find the percent contraction for every test, and then draw some conclusions about what the different solutions did and what might have been the most effective. Okay, and that's pretty much it. Finish your lab report, submit it at the proper length, and I'll see you again next week with a new video.